Does that look really bad to you guys? Looks super grainy. Okay, well, anyways. Hi, my name is Carmen Varela. I am a PhD candidate in Jack Parent's lab at the University of Michigan in Ann Arbor. And our lab focuses on using human pluripotent stem cells, derived models to study monogenic epilepsies. And so my project focuses on SLC6A1, but I'm going to introduce a new human medial ganglionic eminence organoid model that I developed in the lab, and kind of talk a little bit about how I've used it to study SLC6A1's role in development. Okay? Oh, that's not good. Maybe we can just put the one that you have over there. Oh, there we go. Okay, let's try this again. Is that the one you have? Okay, sorry. Got it, okay. So I'm going to talk a little bit, just introduce the medial ganglionic eminence and its importance in development. And then I'm going to introduce the single rosette organoid model that we've kind of adapted. And then talk a little bit briefly about how we generated this MGE-like organoid, but most of the talk will focus on modeling SLC6A1 and an interesting migration deficit that we found in SLC6A1 interneurons. And then finally, I'll talk about future applications and how this organoid model can be used to study other DEEs. Okay, so early on in development, there is a transient region of the brain. Oh, let me see. Oh, there we go. Called the medial ganglionic eminence. It's just labeled MGE here. And that area kind of populates the cortex with interneurons. So most of the interneurons in the cortex are born from the MGE. And the two subtypes mostly that come from the MGE are pervalbumin fast-spiking interneurons and somatostatin interneurons. And the cells that originate from the MGE have been implicated in many disorders, including epilepsy, intellectual disability, developmental delays, autism spectrum disorder, ADHD, bipolar disorder, schizophrenia, and Alzheimer's. So this particular area of the brain is very important for numerous neurological disorders. And so if you aren't familiar with brain organoids or what they are, they're kind of a cellular model that you can use that's an aggregation of brain-like tissue that we can use to study development of cells and how they interact and talk to each other. It's not a kind of whole view of what the brain looks like, but it's a scaled down version. I kind of like to think of it, if the brain is the world, then an organoid is maybe a city. So it can give us an idea of how the cells interact, develop, and talk to each other, but it's still not a proxy for the entire brain. And so there are two kind of main flavors of organoids that are available, as neural organoids that are available right now. There's the multi-rosette method, which produces a lot of these little rosettes that are progenitors in red, and then the blue part is kind of what would be representing as the cortex. But as you can see, there's a lot of variability, and that's a major problem in the organoid field, is variability and reproducibility. And then another method is the single rosette method, which kind of cuts down on that variability. So we decided to move forward with a single rosette brain organoid model. And our model uses first a kind of plating of a monolayer. So we plate cells in a monolayer on the plate. And then at day 4, we use a specialized tool to cut them in evenly sized squares. And then we plate those evenly sized squares onto thick extracellular matrix. So you can see what they look like here. And these squares will self-organize and form these really beautiful single rosette structures, which we can then pick off of the matrix and grow in suspension for the remainder of the experiment. And this is highly reproducible, and they look like this every time. So it's a great model to use, and so we wanted to start with that as a base. Okay, and this is just part of a larger project. I'm not going to go into all this, but another grad student and myself, Miranda Walker, in the lab, we did a massive screening. So we basically tried, when we first developed this single rosette model, we tried some of the standard kind of MGE-like protocols, and they didn't give us very pure populations. So we just kind of decided to start going back to the drawing board, and we tested a ton of factors, including concentration, timing, and mixtures of these different patterning molecules. And then we validated it using single-cell RNA sequencing, comparing it to human fetal development, and then looked at the different cell types, evaluated migration, and finally functional maturation. But what I'm going to focus on right now is the disease modeling that we did with SLC6A1. Okay. So briefly, just to step back right here, the MGE, this region right here, supplies interneurons to the cortex, which they migrate up into the cortex here. And then it also supplies interneurons to the striatum and globus pallidus, which are areas of the brain that are very important for movement disorders. And there are some certain key markers that we are looking for when we want to try to see if we have MGE-like identity. One of the main one is LHX6. Right here, and of course we want FOXG1, which is a forebrain marker, and we don't want any of the markers of the POA or the hypothalamus. And so here we have from our single-cell RNA sequencing at 30 days, we see we have really robust expression of all these markers that are important: FOXG1, NKX2.1, LHX6, LHX8, and GAD1, so this is GABAergic cells. And then we do not have expression or negligible expression of markers from hypothalamus, POA, cortex, and CGE, LGE. So we feel this is a really pure MGE-like organoid. And this is just confirmation using immunostaining to show at day 30 days and 90 days that these the protein from these markers are expressed. And then this is just quickly showing, we wanted to kind of confirm that our cells do kind of model or look similar in gene expression to the human MGE. So we integrated them and compared them to a known published data set, Shi et al., that has done single-cell RNA sequencing on human MGEs. And we can see ours is in yellow here. These are our organoids, and the blue is the Shi et al. MGE, and you can see they overlap really nicely. So that tells us that our MGOs, or our MGE organoids, closely resemble the same gene expression as the human MGE in development. Okay, and so I think you guys probably all know that finding a human cellular model that expresses SLC6A1 in high enough numbers to be meaningful has been challenging, and so we've confirmed that our model does express SLC6A1 very highly in both the 1 month and 3 month time points, and it continues to express as the organoid matures. And so I used this model to study SLC6A1. So I used CRISPR-Cas9 gene-edited fibroblasts, as well as patient-derived cell lines and isogenic controls to test what the importance of SLC6A1 is in early development, because that currently is not known. Okay, and this is just for people who are interested in understanding like where the mutations occur and how they might affect the protein. But I have here, I have all these CRISPR gene-edited lines right here. These are the mutations that were occurred, and here's the predicted truncation site. So we have all of these variants result in a truncated protein, and we have 2 patient variants here as well. And so this is just confirming that mRNA of GAT1 is severely reduced in all of our lines. And one of the things that we didn't expect to find was a migratory deficit, because we think mostly of SLC6A1 as a GABA transporter, not so much involved in migration. But we have an assay that you can plate these organoids onto thick extracellular matrix, and because MGE interneurons are very good at migrating, they like to migrate out into the ECM, extracellular matrix. And so this, of course, is what happened with the wild type and the isogenic control. However, when I looked at the patients and the hets and the knockouts, I saw a severe reduction in this migration. And so we wanted to really understand, is this in fact due to the loss of SLC6A1 or the loss of GAT1? And so this is just the quantification showing a significant reduction in the processy outgrowth as well as the cell body migration, which are both an important part of interneuron migration. So in order to answer the question whether GAT1 is responsible for this migratory phenotype, we did a doxycycline-inducible wild-type expression of SLC6A1. So basically we can turn it on whenever we want in the knockout lines. And I tested two different conditions because I was curious, is GAT1 just important to get the cells going, or is it important through the entire process of migration? So I did two conditions, one where it just briefly got doxycycline, the wild type was only expressed for 4 days, and then I had another one where I expressed the wild type SLC6A1 for the duration of the migration period. And so this is what the wild type looks like right now. We have the TUJ1, TUJ1 is a neuronal marker, so it labels the processes. Here, this is the DNA, but I pseudocolored it in white, so maybe you can see some of the cells coming out. It's a little bit tough on this, but and then this is the knockout line that the wild type gene has not been turned on, and you can see a severe reduction there. And then this is the wild type, or this is the knockout with the wild type GAT1 turned on, and this is the quantification here. And in red right here is, you can see a full rescue of both the processes outgrowth and the cell body migration. But what was interesting was that we also found that this rescue is dose and duration dependent. So when we tried lower concentrations of doxycycline, meaning lower levels of GAT1 will be expressed, we didn't see a full rescue, or we saw partial rescue. But when we used a higher concentration and we used it for the entire period of time, we saw the full rescue. So that tells us that GAT1 is important not only for initiation of migration, but for the entire migration period to be present. Okay, so then we wanted to understand, it's one thing to migrate out into the ECM, but can they migrate like into another cortical organoid? So we did cortical MGO assembloids. We labeled the interneurons in the MGOs with green, MDLX, GFP, and then we just labeled the cortical organoid with red. And you can see here in the wild type conditions, both in the control and the wild type conditions 2 weeks after fusion, you see a lot of cells are already migrating into the cortical organoid. But in both the CRISPR het and the patient line, we see a severe reduction in migration into the cortical organoid. And that's quantified right here. And then so we wanted to understand, is this a cell autonomous effect or a non-cell autonomous effect, which means is this something that's happening intrinsic within the cell, or is it due to environmental changes? And so we kind of mixed and matched the genotype of the cortical organoid and the MGOs. And so you can see in the conditions here where in green, where the inhibitory neurons are wild type, we have really robust migration, regardless of if it's paired with a wild type cortical organoid or a wild type or a patient cortical organoid. But in the two conditions that have either both patient or where the patient MGOs are when the MGOs are patient-derived, we see a severe reduction. So that tells us that this is a cell autonomous effect. And then, so this is kind of just a broad, I don't know if you can read those genes, but this is kind of just a gene expression heat map showing the darker the red, the higher the gene expression is. And these are a whole list of DEE-related genes that I just kind of pulled up to see how much they were expressed in our organoids. So in the different cell types, we have cortical interneurons, we have subpaleo interneurons, and then we have progenitors and glia. And you can probably notice on here you have SLC6A1, which has a lot of expression in the cortical interneurons. We also have SYNGAP here, that's highly expressed, and STXBP1. These are three of the genes that our lab is already studying from this list and using this model, and it's been really helpful. There's actually an STXBP1 poster also at AES this year. So, and then another thing that could take this a little bit further is that our MGOs also produce astrocytes and oligodendrocytes that also express SLC6A1 endogenously. Thank you. So this could be a model moving forward to be able to study these other genes and also these other cell types in SLC6A1. So just to summarize, I've showed you that MGLs closely resemble the human MGE during brain development, and that they produce interneurons with high SLC6A1 expression and also produce astrocytes and oligodendrocytes that express SLC6A1. And that this is a great model for modeling SLC6A1 interneuron migration deficits, which are fully rescued with the expression of wild-type GAT1. And that this migration deficit is cell autonomous. And just kind of stepping back and seeing why is this important. So impaired interneuron migration has been kind of implicated in many DEEs, but this could also lead to dysregulated cortical networks, which could contribute to the pathogenesis of SLC6A1 DEE. And this MGO model could be a useful tool for not only SLC6A1, but for other DE genes, interneurons, astrocytes, and oligodendrocytes, and could be used for a platform to study drug screening, gene therapies, and cell-based therapies. And I'd like to specifically point out my undergrads, 2 of them who are here, who also have posters using this model on SLC6A1, Leah Goldstein and Ria Sanake, and everybody else in our lab and our funding, who we wouldn't be able to do this project without it. And that's it. Thank you.